Somewhere inside your body, a bacterium your ancestors swallowed 2 billion years ago is still working for you right now. It never left. It never fully surrendered. It just changed jobs. Every heartbeat, every thought, every twitch of a muscle runs on power made by a machine that used to be a separate life form. Biology teachers call it the powerhouse of the cell. That's true, and it's also the most boring possible way to describe a hostile takeover that built animals, plants, and fungi. Here's what actually happened. 2 billion years ago, oxygen was a poison. Most life on Earth was small, simple, and built for a world without it. Then oxygen started flooding the atmosphere, produced by early photosynthetic bacteria as a waste product nobody asked for. For most microbes, that was a catastrophe. Oxygen tore molecules apart and killed anything unprepared for it. But a few bacteria evolved. A trick. They learned to use oxygen instead of fearing it, burning fuel far more efficiently than anything that came before. One of those oxygen-eating bacteria met a larger cell. Maybe the larger cell tried to eat it. Maybe the smaller one forced its way in. Nobody alive was there to record it. What matters is the ending. The smaller cell survived inside the larger one instead of getting digested. That should have been impossible. Digestion is not a maybe. A cell that traps prey usually breaks it down for parts within hours. Instead, generations passed, and the guest stayed alive. It kept doing the one thing it was good at, burning fuel with oxygen, far more efficiently than its host ever could alone. The host started depending on that efficiency. The guest started depending on the host for shelter and raw material. Neither side could walk away anymore. You can still read the evidence of that ancient capture inside your own cells today. Mitochondria carry two membranes, not one. A signature of an organism that was engulfed whole rather than built from scratch. They carry their own circular loop of DNA, separate from the DNA sitting in your cell's nucleus. When a mitochondrion reproduces, it doesn't wait for the cell to divide. It splits itself in half, the same way a free-living bacterium does. Even their protein-building machinery, their ribosomes, looks bacterial, not human. That resemblance isn't cosmetic. Certain antibiotics designed to jam bacterial ribosomes can, in rare cases, damage human mitochondria the same way, which is one reason some antibiotics carry warnings about hearing loss. Genetically, mitochondria's closest living relatives aren't random bacteria either. They belong to a group called alpha proteobacteria, the same family that includes rickettsia, the bacteria behind typhus. In other words, the engine running inside every cell of your body is a domesticated cousin of a disease. 4 independent lines of evidence, all pointing at the same origin story. This idea wasn't always accepted science. A Russian botanist named Konstantin Mereshkowski proposed something like it back in 1905, and almost nobody took it seriously. It sat ignored for over 60 years. In 1967, a young biologist named Lynn Margulis revived the idea in a detailed paper and got rejected by roughly 15 scientific journals before one finally published it. Mainstream biology mocked her for years. The theory sounded too strange, a merger, not a mutation. Then DNA sequencing technology caught up in the 1970s and 80s, and it confirmed almost exactly what she'd argued. Mitochondria really did have their own separate evolutionary history, tangled up with, but distinct from, the rest of the cell. One of the strangest ideas in the history of biology turned out to be one of the best supported. Here's why the merger mattered so much. A cell without mitochondria can still make energy, just not much of it. It ferments fuel using its outer membrane as the only real energy-producing surface it has. That works fine at a small size. But surface area doesn't scale with volume. Double a cell's size, and its outer membrane roughly quadruples while its interior volume roughly grows 8-fold. Past a certain size, a cell without internal power generation simply can't produce enough energy to run itself. This is very likely why, for close to 2 billion years, almost nothing on Earth grew bigger than a single simple cell. Mitochondria broke that ceiling. Instead of generating power only at the outer edge, they folded their inner membrane into thousands of ridges called cristae, multiplying the usable surface area inside a space smaller than a speck of dust. Fuel gets stripped apart and fed into a relay of proteins that pass electrons down a chain, like current through a wire. Oxygen sits at the end of that chain, pulling electrons forward and finishing the reaction. As electrons move, mitochondria pump charged particles across the folded membrane, building up pressure like water behind a dam. When that pressure releases through a molecular turbine, it spins, and that spin manufactures ATP, the molecule your cells spend as currency for almost everything they do. A resting adult recycles somewhere close to their own body weight in ATP every single day. Not creates from nothing. Recycles, tearing it apart and rebuilding it, over and over every few seconds. Without that turnover rate, your muscles wouldn't just tire. Your cells would lose the ability to hold their own structure together within minutes. The merger didn't just add a new organelle. It slowly dismantled the guest's independence gene by gene. A free-living bacterium related to mitochondria carries somewhere in the range of a couple thousand genes. The human mitochondrial genome today has 37. Almost everything else got copied over to the nucleus across 2 billion years, or lost outright, because keeping two separate sets of instructions in one cell was wasteful once trust was established. The mitochondrion kept just enough of its own genome to still count as a distinct organism, and gave up the rest of its independence for good. This trade turned out to be so useful that evolution didn't run it just once. A related but separate event happened when a different ancestor swallowed a photosynthetic bacterium and failed to digest it too. That second captive became the chloroplast, the organelle that lets plants and algae turn sunlight into sugar. Two of the most important organelles in complex life started the exact same way: capture, failed digestion, permanent dependency. The trade also runs in reverse. Some modern microbes abandoned oxygen-based energy entirely and stripped their mitochondria down to nearly nothing. Giardia, a parasite that infects the human gut, carries a shrunken remnant organelle called a mitosome that has almost none of a mitochondrian's original machinery left. Other anaerobic microbes carry a version called a hydrogenosome, which makes energy without oxygen at all, using leftover parts of the same ancestral toolkit. Full-power mitochondria, stripped-down mitosomes, oxygen-free hydrogenosomes, chloroplasts, all variations on one original bargain, reused and modified across the entire tree of complex life. Nature runs plenty of partnerships between species. Almost none of them go this far. Corals survive by hosting algae in their own tissue, which supply them with sugar in exchange for shelter and sunlight. But that partnership can break. When water gets too warm, corals expel their algae, turn white, and often starve. The marriage is real, but it's still divorceable. Termites can't digest wood on their own. They rely on gut microbes to break it down for them. But you can, in principle, strip those microbes out and the termite is still a termite, just a starving one. Mitochondria never left room for that kind of separation. There's no version of a complex animal cell that exists for even a day without them, and no version of a mitochondrion, after 2 billion years of gene loss, that survives for long on its own. Every other partnership in biology can still be pulled apart, at least in theory. This one can't. There's a strange rule buried in your own inheritance because of this history. Mitochondria are not shuffled between both parents the way most of your genes are. They travel almost entirely through the egg cell, which means your mitochondrial DNA came from your mother, her mother, and so on in an unbroken maternal line. That's not just a matter of default, it's enforced. An egg cell carries somewhere in the range of 100,000 or more mitochondria. Sperm carry only a few hundred, crammed into the tail for the swim. When a sperm fertilizes an egg, its mitochondria don't just get outnumbered. In most animals studied, they get actively tagged for destruction and broken down within the first few cell divisions. Your body isn't just biased toward your mother's mitochondria, it's actively deleting your father's. Geneticists have used that unbroken maternal line to trace every living human back to a single woman who lived in Africa roughly 100,000 to 200,000 years ago, nicknamed, only half-jokingly, Mitochondrial Eve. She wasn't the only woman alive at the time, and she wasn't the first human. She's simply the most recent woman whose maternal line never broke, all the way down to you. Every other maternal line that existed alongside hers eventually ran out of daughters. Inside your cells right now, mitochondria aren't sitting still like parts bolted to a machine. A single cell can hold anywhere from a few hundred to a couple thousand of them, depending on how much energy that cell burns. They constantly fuse together into shifting networks and split apart again, trading contents as they merge. Damaged sections get tagged and cleared out through a recycling process called mitophagy, and fresh ones get built to replace them. It's less like a fixed engine room and more like a city's power grid, constantly rerouting itself around damage. The partnership has a dark side too. The same electron chain that generates your energy also leaks reactive byproducts that can damage proteins, membranes, and DNA nearby. Cells manage that risk with a constant maintenance program, antioxidants, repair enzymes, and mitophagy clearing out anything too damaged to fix. When that maintenance falls behind, the highest energy tissues in your body feel it first, because they can least afford to run short: your brain, your heart, your muscles. A number of inherited diseases, including conditions like Lee syndrome and MELIS, come directly from mutations in mitochondrial DNA, and they tend to hit exactly those high-demand tissues hardest. Because inheritance runs almost purely through the mother, These diseases pass down a single line with no way for a father's healthy mitochondria to dilute them. That fact alone has already changed modern medicine. In 2015, the UK became the first country in the world to legalize a fertility technique built specifically around this weak point, replacing a mother's diseased mitochondria with healthy donor mitochondria before fertilization. It's sometimes nicknamed three-parent IVF because the resulting child carries nuclear DNA from two parents and a small extra set of mitochondrial DNA from a donor. As of 2025, doctors in Newcastle confirmed 8 babies had been born this way in the UK, all healthy, all clear of the disease, causing mutations their mothers carried. A 2-billion-year-old inheritance rule worked around in a lab on purpose because of a single fact about where your energy supply comes from. Your immune system carries a memory of this ancient origin too, whether it wants to or not. Because mitochondrial DNA still looks bacterial at a molecular level, your immune cells are built to treat it as a threat. Under normal conditions, that DNA stays locked safely inside the mitochondrion, out of view. But when cells are damaged badly enough, in severe injury, for instance, mitochondrial DNA can leak into the bloodstream, and the immune system can respond to it exactly the way it would respond to a bacterial infection, triggering inflammation even though no actual pathogen is present. 2 billion years later, your body still can't fully tell the difference between an invading microbe and the fragments of the one it adopted. So the next time someone tells you mitochondria are just the powerhouse of the cell, you'll know that's the smallest possible version of the story. The real version is a captured microbe that never left, a bargain that built the ceiling for how large and complex life could become, and a piece of ancient bacterial biology that your own body still can't fully call self. It's not living in you. At this point, there's no clean line left between it and you at all.